Hey friends, welcome back to the channel. My name's Rohan. I'm a third-year medical student studying at Cambridge University, and today I'm joined by a very special friend, Bianca.
Hi.
Bianca, introduce yourself.
Hi, I'm Bianca. I'm also a third-year medical student at Robinson.
So today we thought we'd try do like a mock interview just to give you guys a flavour of how things run for like the medicine interviews here at Cambridge. Both of us have gone through that process in different colleges, so hopefully we can give you a little idea about how it goes on. Bianca has something really interesting to tell us about though before we start. Bianca, do you want to tell us about Unlock Biology?
Yeah, so Unlock Biology is essentially an initiative that me and a friend of mine, Aniket Devi, founded a couple of last year or the year before. Essentially, the aim of Unlock Biology is to highlight that biology isn't just about memorizing facts or rote memory. We believe that biology is about things like problem solving, things like critical thinking, actually applying the things that you learn in class to real life situations in the world, things like novel research, because at the end of the day, the students who are studying in sixth form or in secondary school, those are our future scientists, and one day they're going to be the ones that are changing the world, discovering new things. And so the way we test them, the way we engage with them should really highlight those kind of skills. So yeah, what we did is we organised a virtual camp last year. We had kids from all around the UK who participated in it and really got to engage with academics from Cambridge and students like myself, who essentially just to promote their understanding, critical thinking, and they really got loads out of it to show themselves that they too can actually think in the way of a scientist, in the way of a researcher, and they are going to be the future people changing the world. So
that's really interesting, and I've actually looked through some of the questions myself and some of the materials, and it's really well put together, and I'm sure people who went through the programme this summer really benefited from it. So Bianca, where can people find more information if they want to get involved with Unlock Biology this year?
So we have our social media. You can find us on Rohan, I think will link all of this below.
Yeah. Everything will be linked in the description box.
You can find us on Facebook, Twitter, LinkedIn, Instagram, and we also have a website. And if ever you're curious, if ever you want to see some of our material and content, which I think Rohan will probably show as well in this, just email us, just get in touch. And if you want us to get involved with planning to do in-person workshops at schools around the UK this year, just get in touch with us and we'll be so glad to organise something with you.
Brilliant. Thank you so much, Bianca. So now we're going to start the mock interview. So we thought we'd try do like a science-themed one. So this one will be a bit about the immune system. So Bianca will be the interviewer and I'll be the interviewee. So anyway, let's get cracking with the interview.
Awesome. So interviews are meant to be pretty chill at Oxford. They're not meant to be too strict or harsh. It's not kind of like a panel interview, which you might have in some of the other universities where you just kind of fired at questions. It's kind of a discussion or a conversation, because at the end of the day, what they're checking for is whether you can have a conversation with your supervisor or your tutor if you're in a tutorial, because that's kind of the way the learning works at Oxford and Cambridge. So they just essentially tested, are you someone that they'd like to teach or that they'd enjoy teaching?
Yeah, it's definitely not about getting all the answers correct. It's more about how you think and work through problems. So yeah, let's go into the interview.
Awesome. Okay, so first of all, Rohan, you're how are you? How are you doing?
Oh, I'm doing great. Maybe a little bit nervous, but yeah, otherwise fine.
Nothing to worry about. Okay, just to start off, something perhaps you've learned in school. Do you know about white blood cells? Can you name any of the types of white blood cells?
White blood cells. So I know like you have things like macrophages, which are involved in phagocytosis, but you also have things like B and T cells, which are more for the adaptive immune system.
Okay, so you mentioned things like phagocytosis and adaptive immunity. Do you know what these terms mean? Can you describe them a little bit?
So phagocytosis is basically like eating pathogens like bacteria. So the macrophage will come along and will literally engulf the pathogen and then release digestive enzymes to break it up so it's harmless. the other part. So B cells, they are involved in making antibodies, but this is like antibodies are quite specific, which is why they're part of the adaptive immune response. So you can get a more targeted response against like the invading pathogen.
Okay, yeah, exactly. So how about we have a look at what kind of makes an antibody specific? Because you mentioned that antibody is pretty specific. Can you just draw out an antibody for me?
Okay, cool. So an antibody. I think it's like a Y-shaped, like this, and there'll be the heavy chain, which I've drawn, but also like the light chain, which is shorter, like that.
Yeah, awesome. And can you tell me which part is where the antigen binds?
So the antigen binds at like the antigen binding site, which is here.
Yeah. Is that the only antigen binding site? Is there another one?
There's one on the other side as well.
What do you think might be? Why do you think there are two? Why do you think that's in there?
Why are there two? I guess because the antibody's symmetrical, so there just happens to be two. I'm not really sure why there'd be two.
Okay. How about we go into some of the more of the structural details? If you can draw out anything else you know about the antibody structure.
Okay. So this bottom part is the constant region. So FC. Whereas the bit where the pathogen binds to, this is like the antigen binding site, which I think fragment of antigen binding. I think that's why it's called FAB.
Awesome. So do you know any other features on it? What types of bonds do you think there might be?
I mean, I think there is some interaction between the two heavy chains to hold it together.
Yeah. Does that have a name? Do you know?
I'm not sure, sorry.
Okay, so this part over here, this is called the hinge region.
Okay.
And the hinge region is something that just provides flexibility to the antibody. Why do you think that might be good, considering we have two antigen binding sites?
Flexibility. I guess if you have like two targets which are not really next to each other, does this allow the antibody to bend a bit more?
Yeah, exactly.
Yeah.
And even if they are next to each other, like the shape or like the conformation of the antibody might have to move a little bit or change a little bit when it does bind to the antigen. Yeah, that's perfect. So you mentioned the FC region over here, the constant region. Do you know what it does?
I'm not really sure. All I know is that it's not as variable compared to the...
Okay, that's okay. How about we go into looking at what type of interaction an antibody has? So you said it binds to an antigen. Why is that a good thing? Well, why do we need antibodies to bind to antigens?
So like I was saying about B cells, well, the antibodies are kind of specific to like the antigens on the pathogen, which means are you able to target the pathogen more effectively compared to if you didn't have antibodies?
Okay, yeah. So how about you draw out a picture and just a diagram of, say, your antibody bound to the antigen. If you just go down.
Okay. Cool. So if I was to zoom in,
where would this be? What kind of place in the body do you think?
like in the blood?
Yeah,
maybe?
it could be in the blood. Yeah, if you've got a pathogen in the blood, it could be in the blood.
Okay. So just to clarify, do you want me to draw an antibody bound to an antigen?
Yeah.
Okay. So here's my pathogen. Here, like some antigens on the pathogen. And here's the antibody. So antibody, antigen.
Okay, so now that the antibody is bound to the antigen, why is that a good thing? Like, what does it even do? You mentioned that it's specific, but what's the point of it?
does it help? So what I learned is it helps like clump lots of the pathogen or like bacteria
Okay.
together.
So if you keep clumping pathogens and bacteria together, what then? Do you just keep doing that again and again and again? Does that continue?
Does it mean like the pathogen is less active, so less able to cause disease because it can't move around as much?
Yeah, okay, that's one thing that it can't enter cells. It's trapped essentially in one way, because it's bound to the antibody. But even so, if you've got all of these things like immune complexes bound to, so where we've got antibody bound to antigen, or all these antibodies bound to each other, do they remain in your bloodstream forever like that? Have a think
No.
about. Yeah, okay. So why do you think no?
Because, I mean, surely they must be cleared somehow, because surely, I guess, otherwise could the pathogen escape?
Okay, that might be a possibility. And
Yeah.
also, pathogens will be producing things like toxins and things like that. And if maybe you will have the antibody bound to the antigen, but you do need to get rid of the pathogen because at the end of the day, they do cause damage.
Uh-huh.
So we mentioned about the variable region over here that you kind of pointed out. What did you mention this part was?
The FC, so the constant region.
So can you hypothesize a role for the constant region, knowing that you have, from what you've just told me?
So, so I've told you that we have to somehow clear all like the clumping stuff. So,
if you think about what type of molecule is an antibody?
A protein.
Yeah, it's a protein. What is the protein made of?
Amino acids.
Yes. Okay. Yeah. So what is one property of proteins that makes them unique?
They can form hydrogen bonds.
Yeah. Okay.
Yeah.
They can form hydrogen bonds. So a lot of different types of interactions can be formed. Yeah. But why is that? It's because of its shape.
Yeah.
Okay. So they have really specific shapes and conformations. So having a look at that, so where we saw that was useful in the variable region, where you're bound to the pathogen, have a think about what else can be done in the constant region.
Oh, so you're saying it can bind to something specific. Could it bind to, let's say, other immune cells to help clear?
How do you think it will bind to other immune cells? Knowing that it's the protein and it's specific, what do you think it would bind to?
I mean, I guess another specific protein on the immune cells. Maybe like a receptor or...
Yeah, exactly. Something
Yeah.
specific. Yeah, it doesn't always have to be a protein, but yeah, something specific. So if we are binding to, let's draw our immune cell out, and we've got a receptor over here. So we have lots of different types of antibodies. So you have like things like, have you heard of the word immunoglobulin before? It's just a really fancy term for antibody. So you have all these different types of antibodies, immunoglobulin G, M, A, E, and they'll bind to each one of their own receptors. We call these FC receptors. So say if you have IgG, for example, sorry, I've written this upside down.
That's fine.
It'll bind to FC gamma R, so that just means the receptor for that FC region. Okay, so now that you've known that it's bound to a receptor, what then happens? And in fact, what type of immune cell do you think this could be?
Maybe like a T cell?
Okay, so what do you know about T cells? What could T cells do?
So I know there's two types of T cells. There's like a T helper and a T cytotoxic. And the T helper can help bring other immune cells close to a site of infection, but the T cytotoxic is able to like kill cells. Yeah.
Yeah, okay, awesome. So if you had a T cell, for example, here, it's an interesting concept, but T cells don't exactly use the FC gamma receptor as their primary form of communication. We can go into exactly how. One cell that does use this are things like macrophages, like you mentioned.
Okay.
Yeah. So say if we took this as a macrophage, for example, what did you say a macrophage does?
Phagocytosis.
Yeah, exactly.
Yeah.
So how do you think this interaction could lead to phagocytosis, and why do you think?
Could this interaction help create those arms? So like, I think my biology teacher said like pseudopodia, which are able to extend around the pathogen. Yeah. Does it help make the arms, which comes around the...
Okay, yeah. Yeah, perfect. So what will happen is there might be some kind of signalling mechanism inside. You've learned of signalling mechanisms like cyclic AMP going down to something else and causing something, I think. So just a similar sort of thing. There'll be some kind of signaling mechanism that takes place that tells... we call this process opsonization, when an antibody bound to an antigen complex causes some kind of phagocyte, like macrophage, like any other kind of phagocyte, to engulf an antibody-antigen complex. And in that way, you've cleared the antibody and the antigen complex from the bloodstream or from wherever it could be.
Mm-hmm.
And because pathogens are dangerous things, it's important because these phagocytes, as you said, will digest them up, and thereby preventing them from harming our cells and our body.
Oh, so you're basically saying that these antibodies are helping, like, recruit the macrophages, so kind of target the phagocytosis to any pathogens which might be present.
Yeah, awesome. So now that we've digested, so how about you draw out the process of phagocytosis for me?
Okay. So you have the pathogen, which I'm just going to draw like that. And I'm going to draw the macrophage with its arms kind of extending around the pathogen. Just draw a nucleus somewhere there. And then these two arms will fuse until the pathogen is like inside a vacuole.
Yeah.
In the macrophage, and then something will come along to release digestive enzymes into this vacuole.
Okay, yeah, so what you're going to do is that's a lysozyme. It'll release the digestive enzymes and it'll break down the pathogen.
Yeah.
What happens to the pathogen then? Does all of it get digested and broken down?
Yeah, I guess so, because it's completely cleared, or...
Okay, so if it was completely cleared, if our antibodies, our B cells and our macrophages could do all the work, then why do we have T cells?
Do T cells, like... So I know T helper, do they, like, increase the efficiency, I don't know, by recruiting... Are they just like another recruit? Say, like antibodies, they recruit.
Okay, in a way, yeah. So your T helper cells do. So if I just tell you a little bit how a T helper cell works, so say you've got a T helper cell over here. Now T cells, they're a little bit different to B cells in that they can only bind to antigen that's kind of been processed in some kind of way. So not your kind of normal form of antigen that's just bound to a pathogen.
Okay. Yeah.
They need to have antigen that's some kind of processing. So something like a macrophage can do this really well, or any kind of thing like a B cell can even do this. Okay, so this T cell, it has to bind to processed antigen, and it has to do so using something called the MHC. So actually what we have here on our, say, macrophages, we'll have MHC, that's called major histocompatibility complex.
Okay.
It's just a fancy term for basically what presents your antigen over here. Your T cell receptor over here will bind to the antigen, and you might also have another signal over here which we call co-stimulation, so something binding to something else here. So we just call that like signal 1 and signal 2. You need both of those signals in order to activate a T cell. So this can be a T helper cell, for example, and this T helper cell will go on and recruit other cells, as you said. But we have a bit of an issue. That works well and good when we've got an extracellular pathogen, so something that's in the bloodstream, for example. What about viruses? How do they replicate?
So viruses, they're able to like inject their genetic material into cells, and then they can use the host ribosomes to make more viral particles, and when like a critical number have been synthesized inside the cell, they're able to burst and then infect more cells.
Okay, yeah, they do. But if you have a cell here, let's say a virally infected cell that's producing its viruses and they haven't yet burst, can our antibody antigen mechanism work, do you think?
I mean, I'm not sure. Can the protein bind to the DNA or like the genetic material of the virus?
Okay, so you do have proteins that can bind DNA, but our antibody is out here in the blood, because you just told me that B cells will release these antibodies into things like the bloodstream. You also have antibodies floating around something called lymph as well, which is just another fluid within your body. So you have an antibody that's outside. To get to the DNA over here, it's going to have to get inside, right?
Yeah.
Can you do that? Suggest to me, how do you think it might be able to do that, if it can?
So it's a protein. Can it get through like channel proteins in the in like the plasma membrane?
The channel proteins here are a bit too small, say, for a big antibody to go through. And also we've got a bit of an issue because what kind of interactions happen with an antibody? You mentioned hydrogen bonding. Do you know any other kinds of interactions that happen?
So, like covalent bonds or ionic bonds?
Yeah, so ionic bonds. And for ionic bonds to happen, what property does the antibody must have?
There needs to be opposite charges.
Yeah, okay, charges.
Yeah.
Our lipid bilayer, what's its charge?
I mean, the lipid bilayer is mainly fat. Like phospholipids.
Phospholipids, yeah.
So the phosphate groups are negative.
Yeah.
So I guess negative.
Okay, so say we've got heads that are negative, right? What are the tails?
I mean, the tails they're like long hydrocarbon chains.
Mm-hmm.
So are they uncharged?
Yeah, so they're uncharged here. So if we've got an antibody that has a charge and we're encountering an uncharged layer, what's going to happen?
So if it's uncharged, it can't form ionic bonds. So I guess, does it not pass through?
Yeah, it won't pass through. And so can you link that into now what the role of a T cell might be, some of the T cells you mentioned?
T cells. Oh, so are they for being able to, for this type of situation, when your antibodies don't work? Is it like another mechanism of...
So your antibodies are working, it's just that they're not able to get inside the cell in this point because you've got a hydrophobic membrane here. But what we do have are viral particles inside here, and what we can do is we can express them. How do you think they might be expressed? If we think about the system we used in the macrophage, a similar kind of system. That might...
Oh, on those receptors.
Yeah.
Yeah.
So MHC molecules, you can get MHC molecules on all sorts of cells, and any kind of cell can have this. In fact, even tumour cells have MHC molecules. So you have this MHC molecule of a virally infected cell. It can present its viral antigen. So then what do you think will happen?
Well, now that's outside, so can the antibody bind to that?
Okay, so remember what we talked about? Why do you think that actually isn't the case? Thinking about the extracellular pathogen and the antigen that the antibody binds to, you told me that it's a specific interaction between that antigen and the antibody.
Oh, so I guess this can't form the same bonds with
and why is
the
that?
antibody.
Why is that?
Does like the structure change because it's bound to MHC?
Yeah, that's one thing, and also it's been processed as it's gotten to the process of actually expressing on the MHC as we spoke about earlier. So that processing means that it cannot bind to the antibody in the same way.
Okay.
What
do you mean by processed?
Okay, that's a good question. So processing essentially means that what your cell will do is, so some cells, like macrophages, antigen-presenting cells, what they'll do is they'll literally chop your protein up. They might digest parts of it down,
Uh-huh.
and then they'll re-express them. So it'll be a very different protein to the one you initially had.
Oh, okay.
Yeah, so we're expressing a protein here. What do you think can come along? Thinking about the MHC interaction you learned about here.
Oh, T cells, so T helper,
Okay,
or the other.
what do you think?
T
Okay,
cell.
so if a T helper cell binds to this, what will it do? Recruit some more white blood cells, right? That can produce antibodies, right? But those antibodies can't get into the cell, so what's the point in a T helper cell binding? I mean, some
True.
T helper cells will bind, yes, but
Other immune cells, macrophages.
if a macrophage comes along like this, the macrophage was bound. You showed me over here that it bound to this antibody-antigen complex to endocytose it, the phagocytosis, right? That you mentioned.
Mhm.
What can a macrophage do here? Will it eat up the whole cell?
Yeah, because it's a lot bigger.
Okay.
Oh, but this is an infected. It's not the pathogen.
No, it's not the pathogen. It's
So-
an infected cell. The pathogen is inside the cell.
Yeah. So I'm not sure. Would the macrophage still be big enough, or to engulf this cell, or would it not?
There probably are maybe some examples in the literature of macrophages engulfing entire cells, but in this case, a macrophage isn't what will engulf it. Can you think of something else? You did mention the type of T cell.
T cytotoxic.
Yeah.
Yeah.
Okay. So can you draw out the MHC interaction that might happen with a T killer cell?
Okay. So I'm guessing, would it be similar with like a T helper?
Yeah, exactly. The only difference is that a T helper cell, this one's MHC class 2, whereas this is MHC class 1. They're just different types of MHC molecules. And this will bind to the T killer cell. And do you remember what a T killer cell does?
I think it releases some like chemicals which are able to cause the infected cell to like lyse and be
Yeah.
destroyed.
Exactly. So it can cause that, or it can cause apoptosis of the infected cell.
Okay.
And how do you think that might be useful in cancer? Because T killer cells have a very key role in cancer.
Can they target cancerous cells?
Yeah, they do. Why do you think they can target cancer cells?
I guess, do cancerous cells express something on their MHC which attracts?
Yeah, so all of our cells will express something on their MHC. Your skin cells will express something on its MHC. Your liver cell, every cell will have an MHC molecule in it. So why do you think your T cells will attack the cancer cells or the virally infected cells, but not your own cells? Or doesn't that provide a bit of a problem? What if the T killer cells attack your own cells?
Yeah, that would be bad, right? There must be something differentiating an infected cell compared to like a healthy cell. I don't know, from this infection, you said it's this virus particle which is being presented on
Yeah.
MHC. For cancer, I don't know, do cancer cells maybe produce aberrant proteins or something? So there must be something distinguishing a cancer cell from a normal cell.
Yeah, exactly.
Otherwise-
A cancer cell will produce something called a neoantigen. So a neo just means new, so a new type of antigen will be produced. And it literally might look like a normal cell, but actually will have these new antigens which your T killer cells can detect, and it can destroy those cancer cells.
Oh, okay. So basically, cancer can occur if your T killer cells are not working as much, so you're not killing off cancer.
That's an interesting thing. So you're saying that cancer can happen because your T killer cells aren't working that much. That's one possibility. But what tends to actually happen if we look at a tumour we call this the tumour microenvironment, which is basically the area around a tumour. Say this is your cancer cell, for example. It's got its lovely little neoantigen on its MHC. And you have a T killer cell here. So often you do still have T killer cells within your body, because if you didn't have T killer cells, every time you got the flu, you'd die, basically.
Yeah.
Because you need your T killer cells for viral infections. And it's not in the interest of a cancer to kill you off that quickly, is it? Because otherwise it dies as well.
Yeah.
Yeah. Okay, so can you think of another way cancers, you were talking about kind of how how cancer might develop. If we're considering T killer cells, can you think of another way, knowing what you know, I guess, about their interaction?
Could cancer cells avoid T killer cells somehow?
Okay, how could they do that?
So by not presenting something on MHC?
Yeah, how do you think they might not present it?
I mean, or could they pretend that they're just a normal cell? So, because you said, like, even in normal cells, there is something presented on MHC. So could they just present something which looks normal?
Okay, so that's one possibility. You also talked about not presenting something on the MHC itself. So what cancer cells can do is that they can remove the MHC molecules or downregulate the MHC molecules. So in that way, the T killer cell. Why do you think that might be useful, actually?
Remove the MHC.
Yeah, or downregulate them, so reduce their expression.
Why might they? Oh, because then the T killer cells then have nothing to bind to then release their chemicals to cause apoptosis and things.
Okay, yeah. So the T killer cells, they don't bind to the MHC to release their chemicals. The T cells will bind to the MHC, and that will be the interaction, the signaling interaction that takes place. They'll then release their chemicals probably from somewhere else, which will then attack maybe the cell at different areas.
Okay.
Yeah. You might have your T cell binding to some kind of receptor to cause apoptosis, but that's not necessarily the same thing as it binding to the MHC. Okay. Yeah. Exactly. Downregulation of the MHC is one way. Can you think about any other ways?
So I've already said downregulating MHC or presenting something which looks normal. I'm not really sure. Maybe like a different type of MHC, so it doesn't activate the signaling.
Okay, that's a good idea. You also mentioned about T killer cells not being in the picture. Now, we can't get rid of T killer cells around the whole body, but do you think we could get rid of T cells in a local area?
Maybe, but I'm not sure.
Can you suggest a mechanism of how we could exclude T cells just from a local area?
This might sound a bit stupid, but could they be somehow distracted? So could the cancer produce something which makes the T cells go to another place?
Okay, so something that repels T cells.
Yeah.
Is that what you're saying? Yeah. Yeah, exactly. So you've kind of talked about a chemical barrier in that sense, because if it's repelling the T cell away, what other sorts of barriers do you think you could have?
Barriers stopping killer cells coming towards the chemical, like a physical barrier?
A physical barrier. What do you think? What kind of physical barrier? Think about cancers that you know about. What kind of cancers do you know about?
So, like skin cancer or like pancreatic cancer.
Yeah. If we think about it, that this cancer is kind of happening, some kind of tissue. Do you know of any substance that can be good at preventing the T cell coming? Something that's strong, something that's got is inelastic, that could act as a tight rope so that T cells can't enter.
Inelastic.
Yes, inelastic.
So, like the extracellular matrix?
Yeah, what does extracellular matrix contain, if you've heard about that? Yeah.
What
kind of tightrope kind of like material?
Oh, tightrope. Collagen.
Yeah, collagen, exactly. So we have a lot of collagen over here, which basically acts like a fiber. It is your fibrous protein, and your T killer cells get excluded from that area. And in that way, your tumour is protected and it can proliferate and survive.
Oh. Okay.
Awesome. Yeah. Is that enough? I think that was just like a kind of intro to it. I know some of it was probably a little bit more basic. And like maybe towards the end a little bit more challenging.
Brilliant. So should we conclude?
Yeah, obviously a real interview will be very, very, very different. It'll be based on like your kind of style and what kind of level you're at. The interviewer will adjust it. Obviously, I don't exactly know what they teach in school nowadays in terms of. It obviously depends from exam board to exam board, so they will adapt it to what you know.
Yeah. Do you have any feedback for me?
Yeah, so I think towards the end you did pretty well in terms of grasping that collagen is. But I think I did one thing with your interviews. It's really good to sort of say what you're thinking, which is what you did quite well. Some of the stuff towards the beginning, maybe just because they were concepts I would have expected you to know anyway. It would have been nice to sort of go into those a little bit more quicker,
Yeah.
but I think it really depends. Like I think that was really, really well done.
Okay. Cool. Okay, so yeah, that concludes this mock interview. A huge special thank you for Bianca for giving up her afternoon to help record this, and hopefully it'll be a useful resource for you guys. Do remember to check out the Unlock Biology stuff, their webpage, their social media, because they have a lot of workshops and things in the pipeline ready for next year and this year.
Yeah, awesome. Thank you so much, Rohan, for having me. And also, don't take this as like a real interview, that your interview will definitely happen like this. Like, it is very, very, very variable.
Yes. Yeah.
And obviously the level also is very variable. It doesn't necessarily have to be at this level.
Cool. Yeah. Thank you so much, guys, for listening. And catch you in another video.